Hello, dear EHDN viewership. My name is Patrick White. I'm the chair of the European Huntington's Disease Network, and it's my great pleasure to introduce you and welcome you to the first rendition of the 2026 cycle of our EHDN platform meetings. I'm very grateful to Maria Björkqvist from Lund University, who has put together a very exciting lineup of people to discuss the recent developments on a very pertinent topic in Huntington's disease, which is neuroinflammation. And so without further ado, Maria, welcome. The floor is yours.
Thank you so much, Patrick, and thank you to EHDN for making this platform session possible. Accumulating evidence support a role for inflammation in Huntington's disease. The mutant huntingtin protein has been shown to alter immune cell function, both centrally and peripherally. Long before symptom onset, activated microglia and astrocytes can be detected, and levels of pro-inflammatory markers like IL-6 and TNF-α are found to be increased in HD, CSF, and plasma. There are, however, lots of remaining questions, and one question being, how important is inflammation in Huntington's disease? So as Patrick said, my name is Maria Björkqvist, and I work at Lund University in Sweden. It's a great honor for me to chair this platform session where we will discuss and possibly also provide some answers to questions, possibly also provide you with more questions, and focus on if it is time to target inflammation in Huntington's disease. The format today will be, first we will have the opportunity to listen to 2 talks, and this will be followed by a panel discussion, and thereafter there will be a Q&A session. So please note down questions and submit these as we go along. And with this, I would like to introduce our first speaker. Roger Barker is Professor of Clinical Neuroscience at the University of Cambridge and consultant neurologist at Addenbrooke's Hospital. Roger runs the regional NHS HD clinic as well as Parkinson's disease clinics. His research focuses on the heterogeneity of HD and PD and further on how new experimental therapeutics can be utilized. It's a great honor to have you here, Roger, and over to you and to the talk with the title, Inflammation in Huntington's Disease. Is there a case for it?
Thank you very much, Maria. Thank you very much for inviting me. I'm now going to try and share my slides, so it'll be useful if someone just stays on and puts a thumb up. I assume they can see those slides. So if not, thank you very much. So I'm going to give a little talk about why I think there is a case for inflammation in Huntington's disease. I wouldn't say this is an area which I've spent a huge amount of time working on directly, but it's been an area of great interest of mine for a number of years. And this led to us publishing this review a couple of years ago now, written by Sophie Field, who's a wonderful PhD student in my group, and Anna Curl, who was a PhD student postdoc who now works in the States. And this was really provoked by some work we're doing at the moment, which is around a slightly different idea of Huntington's disease, which I'm not going to talk about in this talk, which is to do with transposable elements and what role they may be playing in this condition. But linked to this is the idea of inflammation. And obviously inflammation, as Maria introduced, can take 2 major forms. I'm not a neuroimmunologist, but essentially we have peripheral compartment. So what is wrong in the periphery that may be important in Huntington's in terms of central pathology, and possibly are there peripheral aspects of the disease which we may have overlooked? And then central inflammation, and what role does that play? So obviously we know the gene which causes Huntington's. We've known that for over 30 years now, but we don't have any treatments which actually have worked around that. But obviously, in some way, the mutant huntingtin or the CAG repeat leads to neuronal degeneration. That can activate microglia, which in turn are also expressing huntingtin. And those activations of microglia and astrocytes, especially the work with Steve Goldman showing how the astrocytes can drive a large component of disease pathology, may be contributing. So in this talk, what I want to do is try and walk you through some of the evidence that inflammation is a real thing in Huntington's disease, highlight what we know and what we don't know about it in terms of the clinical significance, and end up saying what we perhaps should do to try and take this forward. So I thought what might be quite useful is to think about Parkinson's. So Parkinson's is the other disease, as Maria said, that I've worked on. I've worked on this for many years. In fact, 40 years ago, I wrote a paper saying that I thought Parkinson's may have an autoimmune inflammatory basis. And the reason I'm going to talk about Parkinson's is this is the evidence which has accumulated over the years by many groups, and not just our group. And Caroline Williams-Gray works next door to me, has taken this on around inflammation to give you some idea of the evidence which needs to be accumulated to help support the argument. So in Parkinson's disease, there is genetic data supporting an inflammatory component in that the HLA region is associated with the risk of getting Parkinson's. We demonstrated that in 2009, and that was subsequently followed up by Nature Genetic papers in 2010 and has been confirmed since. So HLA is associated with Parkinson's, which makes you think that there is an inflammatory component. This is a condition, of course, which is much more frequent than Huntington's, so some of this makes it difficult. Then there is the question of epidemiological data, and there are two studies, one of which is published, one of which is not published, but there's also other epidemiological data around autoimmune diseases in Parkinson's, which has shown that if you look at an epidemiological large cohort, for example, this US population, and you compare Parkinson's versus controls, then actually what's quite interesting is those who are on peripherally acting immunosuppressive drugs actually have a lower incidence of Parkinson's. So it reduces your risk. So there may be lots of other reasons for that. You can't show causality, but there is a link between taking immunosuppressants and reducing your risk of Parkinson's, ergo Parkinson's may have an inflammatory basis. There's pathological evidence going back to the wonderful work of Mayke over 40 years ago, showing reactive microglia in the brain of people with Parkinson's, and that's subsequently been shown by many groups that if you look pathologically in the brain of people with Parkinson's, there's clear evidence of microglia activation and some T cell infiltration. So there's pathological evidence that inflammation occurs in the brain. And then there's some clinical data, of which there's quite an accumulating series of experiments. This is one paper from us where we looked at patients with new onset Parkinson's, looked at their peripheral immune system, and those who had more inflammation in their periphery versus those who had a more anti-inflammatory signature based on a cytokine profile progressed more quickly. In other words, an inflammatory periphery seemed to be driving the disease process. All of those are associations. None of those tell you whether there is actually a causal link. And so Caroline has been one of the first people here to take this on and has done a trial where she gave patients with Parkinson's azathioprine, which predominantly works in the periphery, very well established immunotherapy, which is non-selective. She treated patients over a year, and the paper was recently published in Lancet Neurology, I think earlier this year or the end of last year, showing that whilst it didn't have an impact on the disease progression in the population as a whole, it did have an effect on the female patients in that population. So if we take that as our sort of basis, can we do the same with Huntington's? What is the evidence using these approaches that Huntington's has an inflammatory basis? Well, as you might expect, there's much less convincing data because this is a condition which is much less frequent, and so many of these studies struggle to generate the data that you need. Now, I'm happy to be corrected on any of this, but when looking for genetic data to show that there are associations with HLA, there doesn't appear to be any, although one has to remember that the size of these studies in terms of GWAS are much smaller than ones which take place in Parkinson's. Interestingly, there was this paper just over 10 years ago from Blair Levitt, which looked at this SNP in an HTT promoter, the TNF-kappa B, which actually is also a gene which is very heavily associated with immune cells and immune activation. So whilst the paper was about how this affected the onset of Huntington's through its transcription on Huntington's, it could conceivably be that this SNP was also having effects in other compartments that weren't looked at. In terms of epidemiological data, there is not really very much, but Carsten, who's on the panel, had this very interesting paper where they were attempting to do this, taking people in Enroll, which there are vast numbers, looking at those who had CNS demyelinating disease, looking at disease-modifying therapies in those, and showed that in those patients who had manifest disease, if I've read the paper correctly, those on disease-modifying therapies had reduced progression, at least in terms of some of their cognitive profiles. So there was some, in very small cohort, very small studies, some evidence that using CNS active or drugs that affected inflammation progressing from the periphery into the brain may have had some effect on the disease course. And in terms of pathological data, well, as Maria said, there's quite a lot of data to support this. Many years ago, we were involved with the group at the Hammersmith Hospital in London, where they had the first generation of PET imaging using TSPO ligands, PK95111, and this showed quite clearly microglia activation in the HD brain, and that this occurred in pre-manifest patients, so ahead of any overt disease expression, and that correlated with post-mortem findings. And indeed, there was a very nice paper last year in a different condition in PSP showing that TSPO PET in PSP correlated entirely with pathological microglia reactivity. So there seems to be a correlation between that marker and microglia in these neurodegenerative diseases. And also astrocytic responses and astrogliasis has been well described in Huntington's disease as well. So there is a significant amount of data showing that there is pathological evidence of microglia activation, astrocyte activation. We have embarked on a new study which is around this elements, these transposable elements and what role they may be having. As I say, I'm not going to talk about that, but as part of the discussion today, we have taken some fresh frozen post-mortem brains from patients with Huntington's disease and age and sex match controlled, looking at prefrontal cortex and striatum, and then as a sort of control region, the cerebellum. We've been using nanostring as a way of trying to do bulk RNA sequencing to see what changes occur. We're particularly interested in neuroinflammation and the glial response, so we've used the two panels which are available for nanostring, and we've also done some immunohistochemistry. We're also doing single nuclear RNA sequencing on these samples, but that is ongoing with my colleague Johan Jakobsen in Lund. And what Sophie, who undertook this work, found was that, not surprisingly, the striatum has the greatest, if you like, changes, and that these appear to be around microglia and astrocytes. Again, supporting the idea that the striatum, more than the prefrontal cortex, which is much more than the cerebellum, which reflects the pathology, that there are pathways of astrocyte function and microglia function and activation in these sites. Of course, again, we don't know the direction of travel. Is this a response to the neuronal pathology, or is this actually driving the disease process? And to confirm the transcriptomic data, we've also been doing some immunohistochemistry, which again confirms what many people have shown before, that in the regions which we found the greatest activation at the transcript level, we also see that at the protein level in terms of immunohistochemistry in the cerebellum, which expresses Huntington like every bit of the brain, seems to have no such response. So there's evidence that there is astrocyte and microglia activation. There is evidence in the periphery, and I think Niels will talk a little more about this. As Maria said, these cells express mutant huntingtin, as does every cell. There's clearly evidence that they produce more interleukin-6 and TNF-α. There's elevated soluble CD27 in the CSF. These are often in single studies, although in some of the meta-analyses, if you like, of the studies, a lot of these are very robust, especially the interleukin-6 and the TNF-α. And that is a marker of T-cell activation. And we also had a paper a number of years ago with Francesca Cicchetti in Quebec, where we looked at the blood-brain barrier in patients using 7T MRI, as well as looking at transgenic animals, and showed that there was quite a clear deficit in the blood-brain barrier in patients with Huntington's disease, which then obviously means that peripheral inflammation and peripheral immune cells have greater access to the CNS than one might expect, which makes peripheral immune changes perhaps more significant. And centrally, we see similar things in terms of changes of cytokines and also changes in complement levels. So there's a lot of data supporting these changes. They're often in limited numbers of studies. They're often in limited numbers of patients. So it makes conclusions hard to draw, especially when they're cross-sectional, different stages of disease. But nevertheless, this has enabled people to start thinking about drug targets as ways of trying to look at causality. And that is the real point I want to make. If you want to study the immune system and know what it's doing in a disease, preclinical models are always limited because the immune system of animals is very different from humans. Humanized mice are a nightmare to work with, as we've discovered on other work we've been doing about transplantation of human cells into animal models. It's also very difficult to prove in any of the clinical studies the direction and causality. So that can only be done really by intervening. And a number of trials have been done, which I'm briefly going to talk about. Some have been targeting the periphery, some have been targeting centrally. And essentially, this is the summary of the studies. As you can see, there's a very old study with minocycline, which was a drug that has been used in many neurodegenerative disorders as a way of trying to target microglia, which was out without benefit. There's the Legato HD study, which had a large number of patients who were studied, which, while showing no change clinically, did show some changes radiologically in reduced levels of caudate atrophy. There was a semaphorin 4D antagonist antibody, which was used again without any clinical benefit, but with similar, if you like, imaging findings, reduced rates of caudate atrophy, and some evidence of improved metabolism. There was a small study we did with this drug, which I can't pronounce, which was actually designed more to look at synaptic plasticity in the hippocampus, but has been shown to have some anti-inflammatory. We didn't look at it from that point of view, and the study was actually terminated in COVID. And then there's been this recent interesting study looking at this complement inhibition in a small group of gene carriers. And essentially, as I say, the outcome of these studies have been negative in looking at these drugs. They've all been probably not the optimal drug to look at some of it, particularly in the peripheral immune system. And these two drugs, which we've mentioned in terms of looking at activated astrocytes and this complement inhibition, have thrown up some interesting data, but nothing that is conclusive. I would say the same is true for Parkinson's disease. There's no evidence that any trial so far in Parkinson's disease with anything that's anti-inflammatory has worked. So to finish my talk, I would say, we haven't shown causality. We've shown a lot of associations. We've shown a lot of data. And so I think it remains unknown whether inflammation drives neurodegeneration or whether it's secondary. When I was growing up, all inflammation in the brain was said to be secondary, and that was true for Parkinson's, Alzheimer's, and all of these diseases. I think there's now a real sense that inflammation is a key player. It's not necessarily the initiating event, but maybe a very important catalyst for driving the disease process. We had a look at a lot of biomarkers. I think there has been improvements in the way in which we can assay biomarkers. We now have large patient cohorts. We may be able to probe a bit more to look at that. PET is always limited. There are new PET ligands for microglia, but they bring their own problems around variation in the binding across the population. They're also very expensive, difficult to do, especially longitudinally. And then I think this whole question of, the restaging of Huntington's and where we should be looking at these patients and how we should be looking at them over time, I think is perhaps something we've slightly overlooked. And I think this whole question of the right disease stage at which to try therapeutics is an interesting one. I'm not absolutely sold myself on the fact you have to go very early in the disease process to stop it. But I think there is some logic in the fact that by the time you've lost a lot of cells and you've got a very active immune response, it may be too late to try and dampen down and slow the disease process. But I want to end up by saying, as I said in my introduction, that I think one of the things one should consider doing is whether, rather than doing more animal studies, whilst it's useful to collect data from patient cohorts, perhaps we should be a bit more adventurous and actually do more experimental medicine around many of the anti-inflammatory agents which are now out in the clinic and which we could use to test in Huntington's disease to see whether it really does have any impact on the disease itself. And with that, I will conclude my talk.
Thank you so much, Roger. You have really opened up lots of questions for us to continue discussing during the panel discussion. Now we will move over to our next talk. It's a great honor to introduce Niels Skotte, Associate Professor and Principal Investigator at the Department of Drug Design and Pharmacology, Copenhagen University, Denmark. The Skotte research team focus on clinical proteomics and molecular neuropathology with an emphasis on Huntington's disease, aiming to identify biomarkers and therapeutic targets to support translational development. Niels, over to you and to the talk, Inflammation in Huntington's disease. What can biofluid biomarkers tell us?
Right. Let me just share my screen here. Let's see if this works. And there we go. Do you see it in presentation mode? That's good. All right. Nope. Okay, we'll just swap displays around. So maybe this is better. Okay. All right. Thank you, Maria. Thank you for the nice introduction, and thank you to the EHDN for allowing me to talk today and to talk about something that I'm quite excited about. So I'll talk about inflammation in Huntington's disease and what can the biofluid biomarker tell us. And as Maria said, I'm Associate Professor at the University of Copenhagen, and my name is Niels Skotte. So I think Roger gave us a very nice introduction. I kind of took his overview figure here, and he talked about the innate immune response and how microglia, astrocytes, and complement systems are involved, and how potentially periphery contributes with adaptive immune response, and how this may or may not be secondary, but how there's been clinical trials targeting this. And then he also opened up for there's many different key players involved in the immune system in Huntington's disease. So there are cytokines, chemokines, complement, and a lot of other different proteins. And one of the things I wanted to mention, I took this figure for Roger's paper as well, which is a very nice overview of some of the work that's been done in the past. And I want to focus on just IL-6 because this is one of the cytokines that's been very well studied. And you can see here, if you look across the different studies, that there's some have no change, some show increase, some have no increase, decrease, and increase. And so there's many smaller studies, and maybe the time is now right to actually answer some of the questions that's missing. And I'll try to do that today. I think we agree on a premise. There's inflammation in Huntington's, but now what? How do we move forward? And my job here is really try to fill the gaps. So I'll fill some of them today, and this will be in continuous discussion. But before I jump into the actual data, I want to talk a little bit about what are biofluid biomarkers. So these are molecules that change with the disease. And I also want to illustrate that this is quite complicated because it's not just one molecule at a time. So when you look at a human being, there could be different molecules changing, and at some point we need to integrate those. So metabolites, proteomics, and proteins, so this is what I'll show you today. The transcriptome might change. There could be genetic factors that modify. And then the way we live. So there could be different ways that modifies an inflammatory response. Are we living healthy? Are we exercising, and so forth? So there could be many different factors playing into this. And I want to show you this small video. So this is 8 different types of data collected over 2 years and measured every 3 months. This was during Corona. And what I want you to pay attention to is, for example, the lipidome here. The measurement moves around quite dynamically. The immune cytome moves around quite frequently, but the protein is relatively stable. And so this means depending on what you're looking at, you can get different types of information. And so the dynamics of these different data sets have to be taken into consideration. And I think moving forward, overlaying these and combining them will be the strength for our profiling. So when we conduct biomarker discovery for Huntington's disease, we use different types of biofluids. The work I'll talk to you about today is focused on CSF and blood, but we're also interested in other non-invasive fluids like tears and saliva. These will be collected in a roll or HD Clarity or locally in the clinic that a patient will be associated with. So for this We do use state-of-the-art technology, and this is a collaboration with many different partners, especially the CHDI. But we have to use different technologies to get the right answers. So on the left-hand side here, you can see targeted assays that can be used for NFL or mutant huntingtin. Today, I'll talk about the Astro. So this is classical proteomics, the work we do in our lab. And I'll show you results for Olink and SomaLogic, which are different types of proteomic approaches. So before we can start discussing and looking at changes, what is our standard? What is our biomarker benchmark? And so I think you probably all heard about neurofilament light chain, and you can see on this figure, it's a marker of degenerating or injured neurons in Alzheimer's, multiple sclerosis, frontotemporal dementia, or ALS. It goes up, and there seems to be a small spike around the clinical onset. And when we look in Huntington's disease, you also see this increase in neurofilament light across the different disease stages of the groups. And of course, for Huntington's disease, mutant huntingtin is the cause of the disease, and this also increases with disease. So this is also a nice benchmarker for disease progression. And recently, there's been a strong focus on pro-inflammatory, that's as a marker of medium-spine neurons. And you can see this kind of inversely decreases with disease progression compared to the increase in NFL. So these will be the markers that we'll be comparing to today. So does these biomarkers reflect brain changes? And this is quite important. And yes, the answer is yes, but there's still much more to be learned. But I want to highlight one paper here from Rachel Scarlett's group, where they basically show that the baseline amount of CSF PENK correlate with changes in gray and white matter. And the same they show for NFL and CSF. So both of these two proteins are very good at predicting striatal atrophy. And I think it's very important that the things we measure either in blood or in CSF actually have some meaningful interpretation when it comes to the brain. They also show that both PENK and NFL are very good at predicting and separating different stages. So this is a new staging system here. And you can see PENK and NFL are really relatively good at predicting the groupings between stage 0 and stage 1. Okay, so we know now that NFL goes up, PENK goes down, mutant huntingtin goes up, but what about inflammation? So that's what we're going to focus on today. And so the main driving focus today is that we've been going from smaller studies now to plus 400 participants, and this is even growing in HD clarity. There's even more samples now. This is a very large study to look for biomarker changes. So you can see here the different groupings that we have and how I kind of made them a bit shorter. So we have healthy controls, 2 groups representing the manifest stage, and 2 groups representing the manifest stage where we have early and late, and sorry, pre-manifest stage here. And you can see the sample size of each individual group on the right-hand side. Okay, so to walk you through some of the data today, I have used the CAP score. The CAP score is an estimate of disease burden. So it considers your age and it considers your CAG repeat. And with higher CAP score, you expect the disease progression to follow. And you can see the groupings here, how they're distributed across the CAP score. So the pre-symptomatic HD 1 and 2, and then the manifest groups 1 and 2 here. And just to show you that this makes sense, we have the CAP score going up, and this correlates very nicely with our neurofilament light protein. And you can see here in red, these are the late manifest groups, and down here we have the early pre-manifest groups. So you can also look at the colors to see how this actually fits with the CAP score. So up here we have one that has a high CAP value. So this would be high age, high CAG repeat, and then also the high NFL level. So this means advanced symptomatic stage. And this is a graph you'll see quite a few times moving forward. So why am I showing you data from 3 different technologies? Why not just one? Well, you can see from these figures here that these different technologies measures different things. So many of these proteins are unique to each of these technologies. And we're looking now at several thousands of proteins. The benefit of having the unique proteins is important, but also the fact that some proteins we measure 3 times. So now we can kind of control within this data set that our measurements are correct. And then I want to say that in this data set, we have more than 600 proteins that's associated with inflammation. And of course, we don't have a chance to look at all of them today. So I kind of went back to Roger's paper and focused on his target, a list of inflammatory proteins. Here are the cytokines, chemokines, complement proteins, and other proteins as well. So just before we get going, I want to show you what we call box plots, and this is how the data will look moving forward. And this is neurofilament light across the 3 different measuring technologies. So SomaLogic, Olink, and Astro. And you can see nicely that these measurements are very consistent across the 3 different technologies. This starts with denote if there's a significant difference between the group and the control group. And so you can see this is again very consistent across the 3 different technologies. Olink seems to be a little bit more sensitive here in the earlier stages of the disease and be able to predict the difference between the control and the pre-manifest group. I also want to highlight one thing that is a struggle and something that we cannot fix, because what you can see is that this diversity across HD group 1, you can see that there are some individuals have very low expression and some have very high. And this is consistent across the technology. So this is not a matter of matching or a matter of measuring the protein. This is just biological variability. And this is something we have to understand to make better conclusions moving forward. So what we've done is we've taken this target list of proteins and correlated them with a CAP score. And then what you can see over here on my table is basically the majority of all these proteins that we looked at positively correlate with the disease score. So many of the cytokines are increased when disease burden goes up. The same for the chemokines, the same for complement factors and other relevant proteins like GFAP and C-reactive protein. I'll give you a few examples so you can see how they look, and I'll take you through 4 of these in the next few slides so you can get an idea. So IL-16 is an interleukin. It's a main, it's a chemoattractant and immune amplifier. So it's a cytokine that's involved in immune cell trafficking and communication. But you can see here on the left graph that with increasing disease burden, the amount of IL-F goes up. And this looks very nice. You can see the different groups on the graph as well, but you can also see when you look at the box plot, we only get significance in the last comparison here with the late stage. And this is partly related to when we're looking at thousands of proteins, we are challenged by statistics. So we have very stringent criteria when we do the statistical comparisons. Then I want to show you eotaxin, which is an eosinophil pro-inflammatory chemokine. So this is best known for recruiting eosinophils in the periphery, but is also linked to cognitive decline and brain aging. And so you can see here there is a significant correlation, but it's not as strong as you saw on the other graph where the curve is slightly, the slope is slightly less. And again, here we reach significance at the later stages. Then complement 3, which is a very important key player in the complement cascade. This is a protein that's secreted by astrocytes and is also important for maladaptive synapse loss by microglia. Again, you can see there's a significant correlation across the disease burden and we reach significance at the later stages. And lastly, GFAP that you've probably seen in other publications and other talks, one of those strong markers that we can see in several biofluids. See very strong correlation with the disease burden, and you reach significance at the later stages of this marker. So GFAP is a marker of astrocytic activation or astrogliosis, and as I mentioned, can produce complement 3 that's part of activating or activating the microglia. So one thing I wanted to mention here is, as I mentioned, there's quite a big variability in the expression levels of these proteins. Could it be that some individuals that have higher levels, would that mean a faster disease progression? Could it be that some with lower levels of these inflammatory marker would have slower disease progression? This is something that will be interesting to explore even further. So we know that NFL is a very good marker of disease progression. So it's interesting to see how these markers actually correlate with NFL and potentially with axonal damage. So as you can see in this first plot here, with NFL increasing, PENK goes down. So this makes sense. Axonal damage, NFL is released, PENK from dying MSNs are decreased, and so this correlation is very strong. And so here you have an individual with high NFL and low PENK, and this is so advanced symptomatic stage. So we're starting to get some really nice data from CSF and be able to track disease progression. And this can, of course, be used both for inflammatory clinical trials, but also for other drug modalities. Again, you can see our 16 goes up very nicely with NFL, the same for C3, the same for GFAP, and our chemokine eosin 3 as well. And then lastly, YKL40 that you've probably also seen in other studies related to CSF biomarkers. So now we're starting to get a picture here where we have key neuroinflammatory markers that correlate very nicely with NFL. And we know that we can see NFL changes very early on. So there's a clear trend towards changes starting early and then increasing over disease progression. So I think we can conclude that neuroinflammation is happening. The question is, of course, when, and as George mentioned, is this causal or not? So I just want to keep in mind that correlation here does not necessarily mean causality. And so we're back to the question about the chicken egg, what causes the issues here? And we don't know that yet. We know that they increase together, but what is the cause is still unclear. So just want to sum up a few items before moving on to my last slides. So I've shown you that some of the findings that has been discussed in the past and been shown in smaller studies, we replicate them, we support them. So we see upregulated innate immune proteins, we see complement cascade activation. And importantly, there's also a central peripheral convergence. I didn't show you this data today, but we see some of the same findings in blood and plasma. So the overlapping biology is very supportive. It also means there'll be potential feasibility of blood-based monitoring and not necessarily always needing CSF. And very importantly, the temporal dimension, that some protein changes are detectable before advanced clinical disease stage. This is very important for earlier trials. And then it's also very important, as I showed with NFL, that these associations in neuroinflammatory markers really track with NFL and other neurodegenerative markers. What I showed you today was related to the CAP score. We could also use the Huntington's United Huntington Disease Rating Scale, and we would see the same. So I just want to say across different disease scores, we do have the same findings. So I just want to end on this note on what would targeting inflammation actually mean. And so I want to ask you if you ever heard about this triangle fire. When I was educated as a young man, as a fireman, I learned about this fire triangle. And so to have a fire, you need fuel, heat, and oxygen. And the trick here is if you can remove one side of this triangle, then the fire slows down. So remove one of these three, and that can fix the problem. And I thought about this in the context of Huntington's disease. So if you look at Huntington's disease triangle here, this is something I made up, but it's very clear that Huntington protein itself plays a big part. The CAGVP, the amount of Huntington, somatic instability has been a hot topic recently to explain some of the things that's going on, and maybe inflammation plays a part. And so I think the same applies here. Could you reduce one of the sides of this triangle? You might have benefit. And I also want to make one point clear here. I think that neuroinflammation does not need to start the fire to determine how fast it spreads. So even though it may not be causal, it could still play a very important role. So I think still causality is unclear, but there may be a feed-forward loop where inflammation is either a modifier or an amplifier. So I think there is a strong interest in modifying this. So I want to mention a few things as considerations for future clinical trials focused on immune modulation. So when you consider the treatment strategy, I think Roger also mentioned this, it has to be at the right time, potentially earlier. Cell-specific targeting would be very interesting. Pathway-specific targeting, avoid non-specific immune suppression because there could be both good and bad inflammation. And I think the biofluids might help distinguish and stratify because who has this active immune biology and who might benefit from immune modulation. And with the use of the biomarkers, we could potentially start to do more directed and focused clinical trials. So I'll end here and just thank my team of collaborators, the HD patients and families, and a big thank out to Nesha from my group who helped with the slides, and my previous mentor, Medias Man. And also want to say thank you to Jim and the CSDI Foundation because they funded this work, but also been incredible partners in making this work to be developed. And again, thank you for the HD families and members and HD Clarity sites. All right, I'll stop here and be ready for questions.
Thank you, Niels. There will, however, not be questions now, but a reminder to you all, note them down and submit them in the Q&A. Thank you to both our speakers. This has really set the scene for us, and we will have lots to discuss during the panel discussion, which will now follow. But before following up with the panel discussion, I would like to introduce our panel members. We have with us Assistant Professor Mayke Oosterloo. She's a researcher and clinician at Maastricht University, Netherlands. Mayke has a multidisciplinary approach on innovation in clinical care and research and on translating scientific knowledge into accessible information for the HD community. Mayke is also an expert in tear-based, fluid-based biomarkers. With us, we also have Professor Carsten Saft at Ruhr-University Bochum, Germany. He's a neuroscientist and neurologist with a strong focus on biomarkers, peripheral pathology, therapeutic development in Huntington's disease. His research group has a particular interest in neuroinflammation. With us, we also have Flaviano Giorgini, is a professor of neurogenetics at the University of Leicester in UK. His research group focuses on the kynurenine pathway, which is modulated by inflammation, mitochondrial dysfunction, and regulation of membrane trafficking. And Flav is also currently the science director here at EHDN. In our panels, we also have our speakers. And with that, let's get started. Kinga, could we see each other in another view, or we can continue like this to start off with? So first off, I would like to ask all our panel members a question to get us started. And the question is inflammation an important factor in Huntington's disease? Why or why not? And I would like to direct the first question to you, Roger. What is your opinion on this? Is this important enough for us to work on?
Yeah. Well, you can probably guess what my answer will be given my talk. I mean, I think yes. I mean, as I was saying in the talk, I'm of a generation where in the 80s and 90s, especially when PET came along and people could start looking at microglia, the assumption was that this was all secondary to neuronal degeneration, regardless of the disease. And it's become quite clear in more common diseases like Parkinson's, as I talked to, but in Alzheimer's, that inflammation is an integral part and in the tauopathies. So I can't see why would Huntington's disease be any different in not having an inflammatory component to it. I think the two talks illustrated quite clearly that there are lots of measures of inflammation. Causality is not shown, but it would seem extremely unlikely that it's not contributing to the disease progression. So I don't think it initiates it. I think it is an important contributor. I think one of the reasons we tend to, ignore it in Huntington's disease is that the belief is if it's something's monogenic, the gene is the problem, which is not an unreasonable assumption, and you go after the gene, and that's fine. We haven't been very successful at that in 30 odd years. So why not look for something you can target and treat today on the assumption it may slow things down? And of course, slowing things down is quite important, as my final point I'll make. So we look for cures, which is great, but these diseases typically run a course of probably 30, 35 years. So if you start to develop a disease process in your 25 and 30s and eventually succumb to it in your 60s, but you slow it down by 50%, it'll eventually get you at 90, by which time something else will probably have got you. So slowing down the disease, especially in the early stages of the disease, whilst not curing it, can be extremely effective therapy, which could have really massive implications in how the disease plays out at the individual level. So I think inflammation is very important. I think it is causally involved in some level with Huntington's disease. I think we now have the tools and the agents available to actually look at that. And I think we sometimes have to just not follow where everyone's going around genes and proteins to do with Huntington, but look at other things we could do and which we could do now in the clinic and learn from other diseases which have taken this approach.
Thank you, Roger. Let's hear Niels. What do you say? I really liked your triangle and the 3 different aspects. But we can't hear you.
Still getting used to that, sorry. Yeah, I agree with Roger. I think it's probably not the cause of the disease. And I think also the data I showed you today, I think it's an underestimation of what's actually going on. Because Roger nicely pointed out that some of these changes is happening in striatum and not to the same extent in other areas of the brain. And so if you're imagining proteins being released from a small area in the brain being combined with everything else, and then it's released to the CSF and then the blood, what we're picking up is just a small percentage of the signal that's there. And so that's why I think it's an underestimation. If we had a true readout of what was going on with the striatum, I think these box plots that I showed you in correlations would be a lot stronger. I think the tricky part is here that we know that microglia can be in different states affecting the states of astrocytes, and the whole interplay of these cytokine profiles where some can be protective and in other cases being damaging. It's a very complicated puzzle. And so there's a lot of things going on. So I think my conclusion is it is important, and I think we need to test this. But I don't know what the consequences will be yet. I think that's my stance.
Carsten, what is your view on this? Is
Yes, thank you.
inflammation important?
Thank you, Maria. Yes, I would agree with Roger and Niels, and I also like your triangle needs. And we have some data from the London group, from Sarah Breezy's group, from the young adult study, and they investigated the very early mutation carriers, and they found the YKL-40 being elevated as early as neurofilament. And I think that might be an argument for a very early inflammatory component, and yes, that it might be worth to treat this.
Mayke, do you have something that you would like to add? What's your opinion? Is inflammation important in
Yeah,
HDN?
I think it's very important, as Niels and Roger just showed us. And I think it's also important to remember, I think Roger already said it, we're focusing on lowering mutant huntingtin right now, and that's our main goal, especially in all the trials we've been doing. But I don't think we should aim at one goal. I think we should aim at several goals, and that I believe that we are treating HIV right now, that it may be that we need multiple drugs to try to slow down the progression of the disease, and treating inflammation might be one of the parts that we also need to include, except from lowering mutant huntingtin.
Thank you, Mayke. Very important note. Flav, what's your opinion? How important
I
is
agree with
inflammation?
all the panelists that I think it is important. I mean, I think we obviously have very strong evidence that there is neuroinflammation occurring, with all the beautiful work that was shown by Roger and Niels and others. So that's clear. And I think probably based on other neurodegenerative disorders, I think we have a pretty good understanding that that these could be causative, or at least, contributory. I think in Huntington's we have evidence along those lines, but I don't think it's as robust as I think both Niels and Roger said. And I think that's probably where I think the field would really benefit still from better understanding causation, how this is really affecting it, and trying to find some of these other potential drug targets, as Mike alluded to, that could be maybe in combination quite beneficial to treatment, along with, say, huntingtin lowering or some of these more gene-specific approaches.
But following up on that, Flav, we have talked about we need to do more. So there are knowledge gaps, but what should we focus on? What studies should be conducted? What tools can we use? Are mouse models relevant at all? I heard Roger say that perhaps we should focus on human studies. What's your view on this?
I guess I would think that, well, there really is no perfect model other than humans, of course, and we're a bit limited on what we can obviously do in that regard in terms of mechanistic experiments. But personally, I think that, having a better understanding of the cell types that are important, say, for example, with microglia, and as Niels alluded to, the different kind of microglia that may be involved, I think is going to be really important. And I'm sure other cell types will be quite important to consider as well, such as astrocytes, what have you, but in the CNS. The best way to tackle these, I think there is obviously debate as to the best kind of models to use. And some of this, I think, goes back to timing as well. We're talking about probably an influence on disease that's very long-term and chronic. And are we able to capture that in some of our models, even the mouse models that are somewhat slow onset? I don't really know the answer to what's the best model, but I'd like to see more mechanistic work, say, done in maybe brain organoids, where you could maybe really dissect the different cell types that you're interrogating by different ways, maybe genetic means, and going after certain pathways or targets that way, and seeing, the contribution of certain cell types to the overall pathology, and what the interplay might be. I'm sure that Niels probably has some thoughts on this as well. Saw him nodding a bit, so you may want to add to that.
Yeah, please, Niels.
Yeah, I think coming back to the biomarkers, because the data I showed and also the Roger discussed, much of this is cross-sectional. And I think now within HD Clarity, we are doing longitudinal collections. And I think once we start to get more of this data and we could look at each individual and see how they progress over time, and even within smaller windows, what happens over a week or a month, and then what happens over years, because we need to have a sense of what do these measurements tell us. Let's say you have the flu, NFL goes up, or something else happens. So you start exercising. We don't know how these measurements potentially track. So I think now, and I think it's very exciting to see how many people participate in HD Clarity. And I think there's close to 1,000 samples now. So I think this is a huge resource for the community. And I applaud the patients and families for contributing, because I think this will be the way forward. I think another thing is also like, as I mentioned, we don't know where these markers come from in the biofluids. So they come from everywhere. So if we had technologies that we could pull down, let's say vesicles from striatum, from the microglia or astrocytes, if we could be more specific, we could also be more precise in our answers. So I think this is also something we'll see with technology development in the future.
Roger, you alluded on perhaps the mouse models weren't the tools to go for. What is your opinion here?
I mean, obviously they give you great a capacity to modulate and study and dissect pathways. The question is, how relevant are they for a human disease and with a human immune system? And so I think the organoids is an interesting area, but of course you're only then looking at various compartments, and so you can at least dissect certain cells. So I'm a great fan of patient experiments, actually. And you know, one of the challenges I think in this field is if you think inflammation has a role, then you've got two approaches, I would say. One is you go for a very broad brushes approach. You say, I'm going to give a non-specific immunotherapy and assume if I knock down the immune system, that will help. And I don't really know which component it is. Or you take a much more targeted approach and say, I'm going to give an IL-6 antibody and I'm going to study what effect it has. And you know, one of the ways I would think about this is, you know, you do very small little experimental studies where you're not trying to see whether you actually change the course of disease in the first instance. You see whether it's tolerated in patients, because there are all these antibodies out for all these cytokines. So you could infliximab or an IL-6 antibody. You could say, do I knock it down in terms of a blood cytokine, which you'd expect, because that's what it does. Does it affect the CSF? Does it change anything on the neurofilament? So I just do a little exploratory study. Is it safe? Do I do imaging to see if there's any changes? And you do this in a few patients over a relatively short period of time. Get a safety profile. Have I got target engagement in the compartment, I think, which would be the CNS? And then I go for a bigger study. And I personally would think that these interventions in patients, given we have the reagents available, is the way we should be going. Now, then the question is, well, where do you go? And I would go for early stage disease. I probably wouldn't go for pre-manifest given the nature of the therapies we're given. But the other advantage we have here over other neurodegenerative disorders is these are young, healthy people. So they'll be pretty tolerant of these drugs in a way that if you're going to take mild cognitive impairment or early Parkinson's, it's a much more elderly population. So I don't think we do enough experimental medicine. And I think proper experimental medicine will give you real insight into target engagement, which puts you on the way to looking at how we can modify the disease. And, you know, Niels' talk was an incredible insight in what you can glean from patients in terms of samples. And you don't need many samples, because although there's a big scatter, you can use the patient as their own control if you're following them over time. So I think, personally, I think that's a way we should think about going forward and setting up a sort of mini MAMS experimental medicine platform across the EHDN. There we go.
Niels, did you want to comment on that?
I kind of just want to jump in, because I think also what I showed today was basically just some of the things that we already kind of aware of, we're already tracking. But as I mentioned, we have 600 proteins related to inflammation. And I think it's very obvious now that we can dig deeper and try to understand more on a holistic or pathway level what is going on, because we know from the complement cascade that most of those proteins are changed. So if there's smoke, there's a fire, coming back to the same analogy as before. And then we can start to tease this apart, and with better technology, the timing of it. And I think also coming back to this, that I would like to understand better if there are patients that have higher inflammation than others. And maybe we should be thinking about this, how we develop some of these clinical trials, because if there is no inflammation, an anti-inflammatory drug would not work, but maybe the ones that have high levels, that will where you see great benefit. And I think we have seen industry partners showing some of those ideas in practice as well.
Could I follow up on that, Niels, and perhaps direct to Carsten, Mayke here? Why are there currently no ongoing immune modulatory trials, you think? Thoughts on this? And are there possibly like strategies that we have overlooked? I'm thinking, Mayke, you touched upon, is there a combination of strategies that we should go for? But first, Carsten, would you like to comment on why are there currently no ongoing trials?
Well, I think currently we have this opportunity for Huntingtin lowering therapies, and that's what's going on. And I think that's a big opportunity and an important approach to study. And so that's the ongoing studies. And now we have as an additional component, as Niels showed in his triangle, the somatic instability and expansion, which is also very important and maybe even more important approach. But I agree with Niels and his triangle that inflammation might be another additional component to look for, and maybe we have to have another approach with this. The studies if you look to multiple sclerosis, there is much going on in multiple sclerosis. We have so much development the last 10 years in multiple sclerosis with different immune modulating drugs, and very, very effective. And laquinimod, as the biggest study being performed in Huntington's disease, is not the most effective one in multiple sclerosis. Actually, it's not approved for multiple sclerosis because it didn't show so much efficiency. And in Huntington's disease, we had this big study with 350 or more than 350 participants, but only for 1 year, actually. And it shows some changes in caudate atrophy. So that might be a hint that it's interesting, and motor score might be much more difficult to measure and to over the time. Maybe we need a longer study and a more effective drug. And there are some generic drugs already in multiple sclerosis, which might also be interesting for Huntington's disease, in my view. And I agree with Mayke, of course, that we need a combination of drugs, perhaps, in the future. That sounds reasonable.
Mayke, would you like to comment on this? Why are we not doing this already?
No, I think we've done several trials in the past, but that was before we had the mutant huntingtin lowering trials. And I think then we stepped over to those kind of trials, and we forgot that inflammation was a huge part in Huntington's disease as well. So I think it's just the fact that we have other treatment options that we forgot that we focus on one part, I think, and that's what I said before. I think we shouldn't focus on one thing. We should focus on several treatments at once and try to combine them if possible. And another thing is we've done this in patients who have already disease. And as Niels said that, I think that you want to overcome that inflammation will start at all, I think. And that's also a difficulty why probably these trials in the past didn't work yet. Or maybe we haven't tried the right inflammatory drugs.
Niels, do you want to comment on that?
Yeah, just a small comment, because I think the challenge is also that we've been treating late, and as both Roger and I mentioned, maybe treating earlier is better, but then the question is, what is the readouts? And this is again coming back to brain imaging and biomarkers. So I think what I showed you today is just the top of the iceberg. I think there's still a lot to be done. This will give us some idea of all the things going on, but we might have to develop assays that's been done as for mutant huntingtin and NFL, that's more sensitive and more precise. So it is coming down to the tools to be able to pick this up earlier. Once we can pick these things up earlier in one way or form, there might be a stronger incentive to do some of these trials as well.
Roger.
Yeah, I mean, I agree with everything everyone said. I think we get a bit obsessed with proteins. So I think you have to learn from neurodegeneration. So Alzheimer's, I don't work on Alzheimer's, but personally, if I'd done 40 trials and got 3 hits, I probably would say that I'm not after the right protein, or it's not the whole story. So amyloid and Alzheimer's, I think, is a very good example of where you've concentrated on one thing and actually put a lot of effort into it, and you've moved the dial very little. So clearly there's something else going on there, and you could argue it's tau and it's not genetic, whereas we have Huntington. POG sees synuclein, people have gone after synuclein. And synuclein, so far, that has not worked. And that may be about delivery and right compartment. Now, obviously, Huntington has a genetic basis. It's huntingtin that's produced. So clearly going after it makes sense. But it's not the only pathology you see in Huntington's disease. You know, we talked about inflammation. We did some work on tau. So I think there are all sorts of nuances to this. And I think this reductionist approach going after one protein is a logical thing to do in monogenic diseases. But I think to do it to the exclusion of other things is actually not helpful for the field. And the problem is, once you get into that mindset, like amyloid, that's all that happens. Everybody invests in one therapy and it carries on, and all of these other things. So we could have been looking at inflammation 20 years ago, but we haven't necessarily. I mean, Mayke makes a point, we have done trials, as I said. But actually, I think it's very important to keep broad-minded and see what you can actually do. And I have to say, apart from Niels, is is a very helpful figure. It also reminded me I have to do my fire training. But I think actually the fact that you can target things now, and pharma is going to be working on Huntington and somatic expansions. That's going to be the pharma. We in the academic community can do inflammation, and so why aren't we doing that?
I would like to stop to add a question from the audience here that I think fit the discussion. Do you think that there is or are pathways or inflammatory mechanisms that should be prioritized for further exploration? Where should we go first?
Well, I suppose I could answer that in the sense that I think what we've demonstrated is we don't really know is which is the key pathway. We never will. And it comes back to the point I made earlier about whether you want to do targeted therapies or whether you go for a more broad brush approach, really. I don't think we will ever get to an answer to that question without doing trials, really. I think we can get more data to show that this cytokine is more closely associated with this stage of disease, but it won't tell you whether it's the driving thing. And I think the other word of caution I would always have is the biggest changes aren't necessarily the most significant changes. So subtle changes in a whole combination of pathways, which are hard to detect, may actually be driving a whole disease mechanism, whereas the big change you see is actually downstream and not a causal node in it. So I think I'm sort of avoiding answering the question in a rather political way. If it were me, I think I would probably say which of these cytokines looks the best. So I would, from Niels's data, I would ask Niels what he thinks, and then I would go looking to see if there's an antibody, and then I'll do a trial. If you think T cells are important, then or a B cell, I would go for rituximab or alemtuzumab or one of these antibody therapies, and I would target patients with it. So I don't think we know, but I don't think we'll know until we do the clinical trials.
Anyone else that would like to add on that? Niels?
Can I just follow up? Because I think we also have to be a little bit careful about how we interpret data. Because so now we say NFL is a good marker, and we start to look at other things, but we also have to consider that the stability of these proteins and how they're being released and how they're being measured, just because something is high does not necessarily mean that's the best pathway. So we have to have a little bit more holistic approach when looking at this. For me, I would be interested to see how inhibiting a complement cascade would affect the progression, because we see complement activation in most neurodegenerative diseases, and it both involves astrocytes and microglia, and I think there's some promising data out there that this could be interesting to pursue. And I also just want to come back to the triangle, because I think it's been mentioned. I want to say maybe it's not a Huntington triangle. It could be an octagon. It could be something else, because I think there's many different things playing into this. So I just try to simplify it today.
Flav.
Yeah,
I think it's a good illustration, Niels.
Yeah, I thought I'd use the hand there. I thought I'd just add to what Roger and Niels said. And I agree with both what they're saying. I think we don't know enough really about the best pathway to target, and it could well be that for right now, the best thing to do is to go for a more broad brush approach, as Roger was saying. But I also think that, yeah, I think probably the most promising, if I had to target one, would be what Niels has said, is going for the complement pathway. I think that cascade, I think that could be quite interesting. But I do wonder if if Niels can dig into his kind of information that he has and from other studies as well, maybe one can tease out some mechanisms there. It is difficult, as I said. It's hard to know a lot of times what the signal you're seeing, how important that is. Is it the main player, or is it a more subtle change, as Roger said, that's actually the important player? But I still feel like there's still some mechanistic work that could be done there to find some pathways. But I don't think we really know yet is the answer of what would be the top priority if we were to go for a specific pathway, is my feeling.
Roger.
Yeah, well, I also think we ought to think about the trials which are looking at it and looking at the other parts of the triangle. So, for example, the Unicure, which I don't know anything much about in terms of the data, but you could start to tease it apart. So if you can slow down the disease progression and knock down neurofilament, then what happens to your cytokine changes. Because if it's secondary, you'd imagine you're stopping the cell death, and the cell death will be driving an inflammatory response. But if the inflammatory response is still going on and your neurofilament's coming down, well, then that inflammation seems to be independent of the degeneration. So you can start to work out what might be. So I think we should be looking more consistently across trials at things like inflammation, because that might give you a little bit of a handle into what's secondary and what's primary in driving it.
There are lots of interesting questions coming along now in the Q&A. I will take one here to get us in another angle. The evidence that neuroinflammation is increased in HD is indisputable. However, there is no clear evidence that this is causative. Indeed, maybe the neuroinflammation is a natural protective response in HD. Is there evidence that neuroinflammation in HD is a negative factor, or is it possibly not? So what do you say? There are lots of questions in this regarding this topic. Inflammation a good part or a bad? Or is it a dual? Is it beneficial to start off with, detrimental towards the end? What do we know? Do we know enough? Someone wants to...
I can just say, I think this is a really good question, and I think I don't have the answer, because it's very clear that different cytokine profiles might have different results and outcomes. And as I mentioned earlier, the different cell types can have resting states or different activation states, and that could be different across different brain regions. And so I think we need to look deeper and further to really understand the answer to this question. We can just say a lot of the pro-inflammatory markers are increased, so that would say, okay, maybe it is bad, but there's also some that can be protective. And you don't know if the protective ones are a consequence of the damage and so forth. So again, what comes first? It's a very complicated question.
Carsten, would you like to comment as well?
Yes, thank you. I agree, it's the hen and egg problem, but we know from many mice studies, we know that immune modulatory therapies might be helpful in this mice study, and also from the laquinimod trial, or from our Enroll HD data with Jannis Achenbach study. That's only very few patients. That's not a big basis, but it seems to go in the right direction if you have an immune modulatory therapy, and not helpful to have a high inflammation activity, actually. My guess in that direction. It's no evidence.
Roger, would you like to comment?
Yeah. I mean, I was just going to say that there was some very interesting data last year, which I briefly alluded to, which Mara Mapetti and James Ray have done here in Cambridge, looking at non-HD. This is tauopathies. And it's quite clear that those who do less well, so the people who progress more quickly, it doesn't come answer this question of causality, but it clearly shows those who do very badly have much more inflammation, both in terms of microglia activation and in terms of peripheral cytokines. So TNF-α, and I can't remember, one of the interleukins, were very high in people who do very badly. And at post-mortem, they have a lot of inflammation. So I think, for me, I think neuroinflammation is generally a bad thing. in the diseases that we're seeing. So I can't answer the question is whether there's a good form of neuroinflammation. And I keep coming back to the same point. We'll only know that by intervening. But I would suspect that most of it's bad.
I will take another question for the audience and perhaps direct that to Niels to start off with. Given it's still difficult to conduct early trials where reducing inflammation might have the best chance of affecting disease progression, would the short-term win be looking at inflammation as a response to treatment?
Yeah, I agree with that notion. I think that's part of our work as well. And maybe we could just say, since we have so many proteins, I mean, there's many different things changes at later stages of the disease. It's not just inflammation. There's a lot of things going on. So I could almost build the case for many, and I could expand on my triangle. I think the smartest is to define biomarkers like NFL, YKL-40, DFAP, and now more of these cytokine profiles, that we have a panel that we always measure, and then we see whatever treatment is being tested, does it affect some of these markers? Could it be that we have a treatment that works on something else, but inflammation goes down? What can we learn from that? So I agree that that would be a low-hanging fruit to just kind of keep track of this and see what treatments potentially benefit, and maybe we can then try to learn something about causality or if it's a consequence.
Roger.
I fear I'm talking too much, so you must tell me to shut up. I don't think there's a problem with doing trials. I think there's a perception there's a problem with it, but these are drugs which have been around for decades. So we know the safety profile of these drugs. We know how to monitor. We know how to give them. Every single day in all of our hospitals, there'll be patients receiving all of these immune therapies. So obviously you don't just throw them around, but I think the idea that these are dangerous and difficult to do trials, I don't think they're dangerous and difficult to do. It may be difficult to interpret, and as we've discussed, how you measure everything. But I do really, I don't think there's a problem with doing these type of trials. I think they need to be properly thought through. But actually, I think we have all the tools there to at least start doing this. And I think kicking the can down the road is something we shouldn't do. I think we should grasp the opportunity to do this now. And I think it's going to have to fall to the academics. It's not going to fall to pharma.
Good point to all of us. Switching gear a little bit, complexity. Niels, you brought up that there is this very broad variation amongst control subjects as well as HD subjects when it comes to biomarker levels. There are other complex pathways that affect inflammation. How about aging per se? Aging will affect inflammatory markers and inflammation. How should we consider that? Someone wants to go? Yeah, Niels?
I can start because I also just want to say to the audience that the groups that you saw today are kind of the groups that was part of HD Clarity. So that was the enrollment group. So those matches up when it comes to those groups. And I also want to say, I think we should move away from the groups because you can see within these individual groups, there's such a big variability that we should start to do some of the correlation analysis that I showed you because I think this is more informative. I started talking. I forgot the question. Sorry. The question was...
How can we consider other complex factors that will affect inflammation?
I didn't spend time on it, but I think you could see on the box plot, there is actually a dip with a very early group. It was almost like it went under the control. And they are younger than the rest of the groups. And there are many proteins that just increase with age. We get older, our brains don't function as well. And so a lot of proteins go up and some go down. So age is a huge factor. And this is something we struggle with these in these analyses. And of course, we correct for age. But age is a main factor. And I think if you've seen Henrik Zetterberg's talk, I mean, diet, health, lifestyle, have you been to the moon, or you're smoking? There's a lot of different things that can influence these things.
Another question from the audience. How important are the mentioned blood-brain barrier impairments that were mentioned earlier when discussing inflammation in HD?
I mean, I could probably talk to that. I mean, I don't know is the answer. But I mean, it was a very interesting study that we did. So it's really Francesca Cicchetti that led on it. But I mean, it's clear that the blood-brain barrier in most neurodegenerative disorders is probably not as intact as people think. And obviously quite what drives the integrity of it, there's parasites, there's astrocytes. So certainly in our study, there were it was more permeable. And so there were certainly abnormalities in that. Again, it comes back, well, how significant is that? I think the important point is it means that some of the rules which we apply, that, you know, CNS and peripheral system are probably not quite as it seems. And so I think we need to factor that in, and that has two implications. One is that peripheral inflammation may be influencing central diseases, and secondly, peripherally, drugs which are traditionally peripherally acting may get into the CNS and have an effect there. So a lot of these antibodies don't get into the CNS, but actually, if the blood-brain barrier is not intact, then that might happen. And so, for example, things like infliximab for TNF-α are very effective neurosarcoid, but they're only effective in neurosarcoid when there's a breakdown in the blood-brain barrier. So one of the arguments it wouldn't be useful in disease like Huntington's disease because it couldn't get into the CNS and affect inflammation there. Well, it may be able to do that. And again, coming back to my favored approach of experimental trials, you could actually then measure the concentration of these drugs in the CSF when you give them to patients to see, well, actually, what is the integrity of the blood-brain barrier and how much of this does that get across? And that can also influence how you take it forward. So I think the blood-brain barrier probably is important. Quite how important, I don't know. But I don't think you should assume it's normal, and our data would say it's not. And I think other people have subsequently shown that to be the case.
So do you have anyone else something to add on this? Is there lessons that could be learned from other neurodegenerative disorders when it comes to blood-brain barrier permeability and what effect that might have?
Well, I think you definitely need to know whether drugs get to where you think they're going to get. So I think one of the problems we do is we give things and expect to see an effect. And you need to know, I mean, I had a lot to do with drug repurposing in Parkinson's trials, and you do all these trials based on animal studies, and then you realize that actually the drug never gets into the brain. And you might not need it to get into the brain if you think it's all driven by some peripheral mechanism. But I think understanding how it gets into the CNS is important. And the other thing, which obviously is bread and butter to pharma, but it's a little less clear, I would say, in academia, is target engagement. And the beauty of the immune system, and especially, you know, all of the things that Niels has been talking about, is you can actually look at target engagement. If I'm going to go after TNF-α, do I change TNF-α? You know, you can do that. You know, there are certain things. Am I getting rid of mutant huntingtin in the striatum? Well, I've no idea. I mean, I can't measure it. I mean, I can look at surrogate markers of it, but I can't actually look at huntingtin in the living brain in the striatum with current markers. So, and you know, we look at autophagy in Huntington's disease with David Rubenstein as a therapeutic approach, but can I measure autophagy in the brain? Well, I can't. I can try and look at other things. So inflammation does give us a very unique opportunity to at least look at target engagement. And I think, therefore, looking at target engagement in the CNS and the periphery is going to be important, and is a great opportunity for us to be able to move things forward.
A little bit of another angle here. A question here. From what we know now, could we assume that a better immune system has a positive impact on HD?
I guess it depends what you mean by better immune system, really, because I don't know what a better immune system is.
I don't know. Mayke, what's your thought on this?
Well, I think you can't say one-to-one that a better immune system will benefit the progression of the disease. But I think that if you have a better immune system, you will benefit from it anyhow. I think that counts for everyone who's healthy and everyone with a disorder. So for HD, it will be the same. So if you live healthy, you're probably better off for the longer period, which means that if you have a better immune system, yes, that will probably help you anyway. But in what percentage, I'm not sure to say how.
Carsten.
Difficult to answer. And I think the question is, what does better immune system means? If somebody catch a flu very often, the other one not, I don't know if that's really an influence. And actually, from the few data we have, actually a suppression or modulation of immune system seems to be helpful and not a very active immune system. So difficult question, and question is what does a better immune system mean?
Let's see, we can have time for a few more questions. What are we actually observing in the serum and plasma of HD patients? Which components contribute specifically to peripheral inflammation versus neuroinflammation? Do we know? Does the inflammatory profile seen in CSF align with what is detected in blood? Niels, does it align? Is it the same pattern?
What's right? Go ahead.
Is it the same pattern in CSF and in plasma?
Yeah, I showed it very quickly in the table in the beginning that a lot of the things we do see in CSF is reflected in plasma. But again, CSF is just a way better fluid because it's surrounding the brain and it's a closer surrogate marker. And I think once your proteins, if they come from the brain into the blood, it gets very much diluted. So if it is a protein that is also expressed in the periphery, you may not be able to pick it up. So it's a very small signal. So I think changes in, NFL is increased in blood. It's a very good marker, but I think a lot of proteins, you almost need to tag them to say this is from the brain and this is from the periphery to be able to interpret it. And I think I can also say we have mouse studies where we try to look for changes in the brain and correlate it with CSF and blood. And there's some proteins that might go up in brain but go down in CSF. So it's not one-to-one correlated. You have to be careful sometimes when you interpret the results of the biofluids. What we're showing today is very well validated, but there's still a lot to be learned.
Roger.
Yeah, it's a bit tangential, but one of the things we've been looking at Parkinson's, I know it's not Huntington's, but we've been looking at these transposable elements in Parkinson's, and what we found is that if you take a brain, the pathology and inflammation is different in different parts. So within the basal ganglia, for example, you will see that there's increased transposable elements and increased interferon response in microglia. But if you look in that same brain in the amygdala and cortex, there's a lot of alpha-synuclein pathology, but there is no microglia interferon response. And I think the significance of that is, there are some challenges not only between the blood and the CSF, but is the disease process the same in every part of the brain? So actually, are they all following the same course, or is it different in different parts of the brain? Now, if that's the case, obviously in our study, it could be that it's the same disease process and we've caught it at different times and we see different inflammation at different stages, which comes back to this question about the timing, and inflammation may actually change during the course of the illness. And that has been seen in other diseases. I think alpha-synuclein antibody is much higher in early PD than later disease. So the disease process and inflammation may change temporally in Huntington's, which again, I think creates further challenges. So I think, and so I suppose what I'm making is there can be temporal changes and there can be changes. It may not all be the same disease in every place that it strikes, really, and that creates complications for it.
There is one question that I really need to ask before we run out of time. So let's take that so we don't forget it. If we are thinking about today's discussion, what would be the recommendation to HD patients today? Mayke, would you like to start us off? Would there be a recommendation, and what?
I think we're allowed to say that inflammation plays a large role in HD and that we should not forget about that. I think we can tell the patients that. I think we should say that they should use non-steroid inflammatory drugs like COX-2 inhibitors or things like that, because if they start reading on the internet about that, they might come up with this. And as I just said before, that starting new trials regarding inflammation would be something we should consider. And I would also say that it's very important that we keep looking up in different angles in how we can try to treat the disease and try to decrease progression. And that might not always or maybe alone be the lowering of the mutant huntingtin protein, because I think that patients nowadays believe that's the holy grail. And as I said before, I don't believe it is. And we have already seen that some of the trials failed or had some success, but well, I think we have a look in a broader perspective, and I think we should tell our patients that as well.
Someone else that would like to add? Carsten, please.
I think it's too difficult, and I think we follow all this discussion and the fantastic talks from Niels and Roger that we don't know enough to really make a recommendation taking something for patients. My recommendation would be to participate in Enroll and HD Clarity to learn what's going on really. And perhaps we can also learn if somebody takes immune modulatory treatment for other reasons and he participates in Enroll, we can also perhaps learn participants if there are enough, something like that. So that would be my main recommendation.
Roger.
I mean, I would echo that. I think trying to encourage people to get involved with research. I think we need to be creative about picking out the patient populations, as Carsten tried to do in that study. You know, people who are natural studies, if you like. And I would tell patients not to take anti-inflammatory drugs on the expectation it might help them, because of the side effects of non-steroidal anti-inflammatories are not insignificant. I mean, we might get some data from aspirin, for example. I mean, I know it's a very, but you know, what happened, because there will be significant numbers of people, and I would thought, on enrolled HD who are on aspirin. And do they follow a different course to people who are not on aspirin? And, you know, so there could be some easy wins with just looking at databases that we've got already. But I would agree with what people have said before. I wouldn't encourage people to take non-steroidals if they come to clinic and ask. Healthy lifestyle, that's what they need.
Is there anything that we should bring up that we have missed? We have a few more minutes. Is there any? I mean, we have stirred a lot of questions. I'm not sure we have provided so many answers.
Can I ask you a question, Maria?
What do you say?
I'm going to ask you, what do you think? Do you
What
think
do I
information is
think?
important? Because you've asked us all the questions. So I want to ask
Yeah,
you,
I
what
think
do you think?
I think that we still have lots of studies that we need to conduct. And my opinion is that I think that we might have overlooked something that is accessible. Lots of these treatments are out there, and as a combination with other approaches, I think that there might be something here. So I'm really happy that you all have provided so much questions and knowledge, and hopefully indicated a lot of knowledge gaps for the audience to pick up. We need more studies, and we need more clinical trials. And I think, Roger, it was really good that you put us off in a direction that we from the academic side, we should really take a lead here and not be afraid to start off on these, embark on these. Patrick, would you like to say something? We have 1 minute to go.
No, just thank you, everybody, for a fantastic session. I cannot provide the final answer for what role inflammation plays. I think Carsten made an incredibly important point in that we need the participation from the patients who are fantastic and already in volunteering, and clearly inflammation is a frontier where there's a lot of progress to be made and to be expected. So I'm very, very happy, Maria, that you put together this great panel.
Thank you, Patrick, and thank you to all of you that provided us with so much knowledge and questions. And thank you to the audience out there for hanging in there and asking questions. We didn't answer all of them. We did answer some of them, but we hope we stirred some interest for this topic. Thank you.